Gastrointestinal Microbiome by Mackenzie Mickelson for Black Hills State Genetics. Within the last decade, research regarding the gastrointestinal microbiome has flourished. What was once thought to be just a digestive organ now is thought to hold the key to information on optimal health and causes of disease. The food industry has adopted this new knowledge to market a plethora of food and supplemental products. These probiotics, or fermented foods, are plentiful on shelves and in advertisements claiming to improve health. The gastrointestinal microbiome is a major factor in metabolism and energy regulation. This fact is a large reason why the food industry has created a fad to sell these gastrointestinal microbiome products. Trillions of microbes inhabit the typically healthy person. Microbes do not always inhabit the human body, though. Babies are born sterile and are immediately colonized by microbes upon birth. The way the baby was delivered will affect the microbiome that they acquire. If the baby is born vaginally, lactobacillus and prevotella will be more prevalent because these microbes are found in the vaginal microbiome of the mother. If the baby is born via cesarean, the baby will have more skin microbes such as Staphylococcus, Corneobacterium, and Propionibacterium because these are found on the skin. The gastrointestinal microbiome composition is determined by many factors, with the main one being diet. Although the gastrointestinal microbiome is rather stable, there's always a constant flux of bacteria entering and exiting. The microbes that enter are from the environment, food, and oral cavity. Other biochemical factors that determine what bacteria is in the microbiome are the pH in different parts of the body, oxygen, antimicrobial peptide gradients, also known as AMPs, the presence of bile acids, and the speed of transit. The pH is lowest in the stomach and gradually increases as you go through the gastrointestinal tract. This is the opposite of oxygen, where oxygen is the most abundant at the beginning of the tract and decreases and becomes anaerobic towards the end of the tract. AMPs in different parts of the gut suppress certain bacteria. The higher the AMPs, the faster the speed of transit. This is useful when the gastrointestinal microbiome is infected. If there is a high concentration of AMPs, the transit speed will be faster, thus the microbiome can become healthier in a quickly manner. We've been talking about how microbes get into the microbiome, but what is the microbiome consisting of? As previously said, trillions of microbes are in the typical healthy microbiome, and these are mainly in the colon. The microbiome is made up of different types of bacteria, archaea, microbial eukaryotes, and viruses. Bacteria from the phyla, Bacteroidetes and Firmicutes, dominate the microbiome of a healthy individual. Additional phyla of Proteobacteria, Actinobacteria, Verrucomicrobia, and Fusobacteria are also present in low concentrations. Stool sampling is the most common sampling of the gastrointestinal microbiome, but this usually shows more of what the concentrations of microbes are in the colon. This sampling has shown extremely high densities of bacterial families of Bacteroideaceae, Prevolotellaceae, Reichenelliceae, Lachnospiraceae, and Ruminocoaceae. Separate testing shows that the small intestine is enriched with Lactobacilliceae and Enterobacteriaceae families. The Human Microbiome Project, also known as HMP, In the Microbiome Genome-Wide Association Study, also known as MGWAS, are tools researchers have been using to identify important genetic variables that affect the gastrointestinal microbiome. Early MGWAS identified 83 associations between the microbiome and the human loci. More recent research has indicated an association between LCT, which is a lactase gene, and the abundance of bifidobacterium, which is from the phyla Actinobacteria. The gastrointestinal microbiome has been linked to several diseases, and cardiovascular disease is one of them. G et al. Ran a study comparing metagenomic shotgun sequencing on fecal samples. These samples came from 218 individuals with atherosclerotic vascular disease, also known as ACVD, and were compared to 187 healthy control samples. Abundant differences were shown in major genera of the gastrointestinal microbiome between the two groups. The ACVD group showed a relative reduction in bacteroides and Prevotella, but they also showed an enrichment in Streptococcus and Escherichia. Gestational diabetes mellitus, also known as GDM, can also be affected by the gastrointestinal microbiome. The gut microbiome is a major factor in metabolism and energy regulation. Nearly 10% of our daily consumed calories are provided by the gastrointestinal microbiome. This is partly via the degradation of complex polysaccharides into short-chain fatty acids via fermentation. Therefore, the gut microbiome is a major factor contributing to obesity and thus linking to type 2 diabetes mellitus. In a study on GDM, experimental animal models found reduced numbers of bifidobacteria led to enhanced organs producing liposaccharides, endotoxemia, which is a low-grade inflammation because of increased circulating endotoxins, and also associated obesity with insulin resistance. Also, the biosynthesis of branched-chain amino acids, insulin resistance, and glucose intolerance have been associated with species in the gastrointestinal microbiome such as Prevotella copri and Bacteroidetes vulgates. Helicobacter pylori, more commonly known as H. pylori, can also infect the gastrointestinal microbiome. H. pylori is a bacterium that has adapted to the human host. Coevolution of the bacterium with humans has occurred due to the restricted host relationship. This has also shaped the molecular determinants of host pathogen interaction drastically. H. pylori infection leads to chronic gastritis, ulcers, and subsequently gastric adenocarcinoma and malt lymphoma. Some benefits have shown to be associated with H. pylori infection. An example of this has been shown in individuals that were infected early in life. This could significantly lower the risk of those individuals developing asthma and celiac disease later in their lives. Many of the beneficial traits are induced by immune system modulation of H. pylori due to the induction of tolerogenic immune state, which is the induction of regulatory T cells. This helps the bacteria to persist in the human host. The gastrointestinal microbiome is a microbiome that has recently gained more attention in the last decade. It contains many microbes that work together and affect the overall health of a human. Diseases can be affected by the gastrointestinal microbiome, such as ACVD, GDM, type 2 diabetes, and obesity. Different concentrations of microbes can be utilized to tell the difference between healthy individuals and diseased individuals. Genetic research is being conducted to find links between genes expressed in the human genome and microbes found in the microbiome. This can be improved by using more mGWAS and the HMP to continue learning about the link between the human genome and the gastrointestinal microbiome. This can also be utilized in the medical field. But this research is relatively new and has a lot to find. Thus, the gastrointestinal microbiome is still a relative mystery to science and the medical field.